So the masters are for us the main tool we have in order to create human capital. We are involved in 8 masters. We try always to be a partner with very prestigious institution in Spain, in this case with the University of Lleida, and we try to invite 2 other institutions to allow us to have a very solid and strong partnership and consortium in order to offer the best high-level training that can be offered in any particular topic. So thank you very much, not only to the people that are today with us, but to all the participants, all the teachers and speakers and lecturers that participate in these masters. I invite you, all the participants in this seminar, after the seminar, I hope that you will want to know more about our masters, our specialists, to consult, to visit our website. And I hope some of you are going to join our master. And maybe I would like to join you to meet you here in Zaragoza at the end of September. Okay, so thank you very much. I give the floor to the rest of the participants. Bye.
Thanks indeed, Raúl. Thank you very much for your words, okay? So we start with Elena. Elena, the floor is yours, okay?
Thank you, Ramsey. So today I'm gonna give you a very quick overall look at what we do in our lab. And our lab is, as you can see here, is in Madrid, in Spain. It's a joint center. Institutions, 2 institutions participate in this Center for Biotechnology and Genomics of Plants. It's the Polytechnical University of Madrid, where I'm a professor, and the CSIC. So this is a latest picture of our lab members from this Christmas, and this is a research line where 2 principal investigators collaborate. It's Juan Carlos del Pozo and myself. And what I'm going to be talking about today is basically one main line of research that we've been following during the last 3, 4, 5 years, in which we have joined both our expertises in order to try to obtain more information about how can we make better variants of plants that would take a more resilient survival in case of climate change. So Juan Carlos is an expert in root development, and he's been working for many years in how the roots develop whenever they have a phosphorus deficiency or the other abiotic stresses, mainly heat. And I've been working on epigenetic regulation of gene expression also for many years. So because in climate change, this is a situation that the plants are going to find more and more often, like phosphorus deficiency, we should be using less and less fertilizants. So this will be the situation more and more often. And also, as you know, we're going to be finding more and more heat waves. So we know that there are some epigenetic mechanisms that control the adaptation and the response of plants to these situations, but it's not very well understood. And we wanted to study really what was going on there, which is the first step, in my opinion, to be really later to develop an action that will create plants that are better adapted or whatever. You first have to understand how the mechanisms work. So I was just looking a little bit, and from the classes that I teach in our university, I usually present it like that. What are the hottest things now that we want to do now in the breeding programs, or that the agriculture people ask the scientists for? Well, we all want plants that behave better in this scenario of climate change. So this is where our research is situated, in understanding how plants behave in the case of extreme temperatures and poor soils. And because it is always also going to be very important to minimize the fertilizer use, which is not only good for the farmer, but it's also good for the plant. And also, when we talk about the new developments in breeding or the new techniques that have been incorporated in these last years, well, of course, we're going to be talking today, I guess, about CRISPR-Cas and other new technologies that have become very hot these last years. But what I'm going to be talking about is located in one of the newest and more cutting-edge technologies, which is the epigenetic modification in order to create more variety or new varieties that can behave differently. So it's not, our last aim will not be just to modify the DNA, but to modify the modifications that accompany this DNA in order to produce plants that behave differently, so the genome won't be edited at all, or of course, transgenic. So just to very, very briefly, but to situate yourselves in and try to make you understand what we work with, what we do is to try to look at in vitro and model plants, but in situations as close as what the crops actually find in the field. So for that, we have a bit of a complicated experimental setup in which we differ from the way that the people usually working with Arabidopsis with model plants in vitro systems work. So we always keep roots in the dark, and when we apply increased temperatures, we don't apply increased temperatures to the whole plant, because that is not what is happening in nature. In nature, the plants have their roots growing within the soil. So even if in the environment it's 40 degrees or whatever in the summer, the roots are never going to be exposed to these very high temperatures, because just the soil around them creates a gradient, and they are always exposed to lower temperatures. So we mimic these situations and then we study the effects and the role of epigenetics on this adaptation of the plants to the new behaviors. This is just to show you that in the regular ways of studying heat responses, when you apply high temperatures homogeneously to the shoot and the root, you see these kind of responses, but that is not actually what is happening in the field. So when you have your roots growing in this gradient, you can see that they are able to develop normally and even better than normal roots. And this is actually why we have plants that are growing at 40, 45 degrees in the summer in Spain or in all the Mediterranean countries, which would not be possible if that was the response actually happening. So our experimental setup allows us to study really the things that are actually happening to crops when they are growing in the field, or we are closer at least than we were before with other simpler experimental setups. So in our experiments, as I said, we study how the plants grow in phosphate deficiency and how the plants grow at high temperatures, and then the combination of both these stresses. And one of the first things that we've been able to see is, for example, I'm just showing you here some of the results we've obtained, is that there are a lot of pathways that are controlling the response of the plants to these stresses. But something that is very important, or at least that control a lot of the genes that are expressed or not expressed in response, so that the plant respond to these stresses, are the microRNAs. And we've been able to see clearly how either in both tissues, root and shoot, we have a very common set of microRNAs that are responding to the different stresses. And then we have also specific stresses that respond to one stress or the other, or the combination and stuff. And this, for example, we have found a very interesting microRNA that is only induced in the root, but we can see later that it moves through the xylem and reaches the shoot, where it's actually producing an effect. And this is something that we are now in the middle of studying, how maybe we can introduce some changes or control how the way these microRNAs work, because if we create varieties with these changes in these microRNA workings, then a lot of genes are going to be changed in their expression because of these master regulators. And this is a project now that one of the students of this master is actually working and has been working this year, and we are very happy she's making a lot of progress, and soon we'll see the results published. Another aspect of epigenetics that we've been studying in this context of stresses is, for example, DNA methylation. We've seen that when the plants are subjected to different stresses, their levels of DNA methylation change, but they don't always change the same, and not all the different types of DNA methylation that we can see are always changing the same ones, but it depends on the type of stress that we get a response or another different response. So I'm not gonna get into the details, but the control of what nucleotides are methylated or not is something very complex in plants. Is the organisms with the highest complexity in terms of DNA methylation. We have pathways that are specific of plants and don't exist in any other kingdoms. And so after lots of experiments and a lot of work done here, we think we have identified that basically most of the response is what you can see here in blue, that happens after heat and phosphate deficiency is depending on this protein that we have here, CMT2. And in fact, when we look at the mutants in this protein CMT2, we can see that they develop much better in the presence of stresses. So I don't know if you can appreciate it here, but when we quantify, we can see that the roots of these mutants, when they are faced with the stresses, they are longer and they have more lateral roots. So they have a stronger root system that just make that the plant survive better and produce a lot more in the presence of these stresses. So now we are think that doing for example, a CRISPR-Cas or mutating this gene in crop varieties could be interesting because it would produce plants like this that respond better to the stresses. But also we've opened this door that I was talking about at the beginning that is very, very new. And I think we are very excited to be having the first ones having candidate places to look at, in which we can try to control the methylation in some certain places where CMT2 is put in methylation. So we can just remove the methylation, not the whole CMT2 gene, but one of the sites that CMT2 is methylating. And we think that we could see the same effect, plants that develop better. And this is something that we are now trying to put into the table so that the new project will be directly to try to do that and in model plants and then in crops to see if this is a way to generate new varieties that would develop better. And that's it. That's all I wanted to tell you about the work we do here at the CBGP.
Thanks indeed, Elena. Thank you very much for your presentation. It was very huge. Okay, thank you very much. Now I give the floor to Diego. Diego, the floor is yours. Go ahead.
Minimize this. Okay, thank you very much. Hello everyone. My name is Diego Orzáez. I work in the IBMCP, it's an institute from CSIC in Valencia. And I will give a general vision about my views and some of the work we do around new breeding techniques, and in particular CRISPR-Cas. And my intention in this short talk is just to give you, as I said, a vision of how is the situation now regarding new breeding techniques. Let me see if this moves. Okay. One of the reasons clearly that we need these new breeding techniques is because we need breeding, we need genetic breeding in order to improve our crops and to face the challenges. Here I put a couple of slides of graphs that show that we really need breeding in order to cope with the future or present problems that we are facing in agriculture, from pest and disease resistance, improvement against environmental stress, acceleration of new crops, or biofortification. And here a couple of graphs that show that most of the gains in productivity and other aspects that we have been achieving in the last decade are mostly due to breeding. So breeding is really what we need in order to cope, to survive in the future of agriculture. And then what are these new breeding techniques? These new breeding techniques are alternative breeding techniques to the classical breeding tools that we had that help us to get plant varieties faster and probably more efficiently, and especially with greater precision. And that, contrary to the traditional transgenesis, do not use external DNA sequences or at least sequences that are outside the breeder's pool. And basically, the most well-known breeding technique is CRISPR mutagenesis that I assume most of the audience have listened, do already know. But we also have, I would like to take some time to talk about a little bit more advanced breeding techniques like base editing, prime editing, and cisgenesis and intergenesis that are alternative also new breeding techniques that are also considered within the basket of what are new breeding techniques. But let's start from the beginning. About the CRISPR mutagenesis, as I said, this is the most commonly used now, more than 10 years. That is technique is our hands. It's an amazing technique in which we are able to produce breaks, double-strand breaks in the DNA, in the genome of the species in a very precise position. We can design exactly which part of the genome we want to introduce these double-strand breaks, and as a consequence of that, the cell machinery repairs this double-strand break and produce a change, a mutation, and this mutation can have a breeding value. It's the equivalent of changing, making small changes in a text, right? And this is the reparation can work in two ways with a homologous recombination, that it doesn't work very well in plants. But this is a non-homologous end joining system, but that is the actually that works very well in plants and produce these single nucleotides or small deletions or small changes that contribute to these valuable breeding changes with value in breeding. For the last 10 years, we have been working in our laboratory in developing these techniques and applying to plants. We have several examples, but I choose two of the early ones that I think it illustrate the use in breeding for this of this system. Most of them have to be with organoleptic traits or visual traits that can be very easy followed very easily. One example is this green flesh mutation. You know that chlorophyll in tomato declines upon ripening, and if we mutate with a single nucleotide mutation that breaks the one enzyme, one gene that is involved in degradation of chlorophyll, then we have these brown tomatoes that you can see here in the slide, and that let me show. And that are very easily to obtain using these CRISPR-Cas systems. And also another example, we also with eggplant, you have eggplants, you know that they have a problem with browning, and it's relatively easy to break down or to mutate some of the genes that are encoding polyphenol oxidases, and then reduce the oxidation associated with the cutting of in fresh eggplant. So this is just simply a few examples of single mutations, but I think one of the most important applications that it could have also the use of this mutagenesis, CRISPR-based mutagenesis in terms of breeding is when we are able to produce multiple mutations in a single plant. You know, many crop plants have gene families, and those gene families normally cannot be mutated, all of them, all the elements of all the components of a gene family at once using traditional breeding techniques. But we can use these CRISPR-Cas mutagenesis approaches in order to simultaneously knock out many of the genes. This is a beautiful example that we have here in Spain of Francisco Barro, who mutated many of these genes called gliadins that are responsible for gluten in accumulation in wheat, and the removal of some of these, many of those gliadins have, as a consequence, a low content of gluten in wheat. This is a beautiful example. We also apply some of these multiple editions or multiple mutagenesis in breeding plant biofactories. In our case, we're interested in tobacco plants that can be used as biofactories. And here you have examples of field trials of tobacco plants in which we have completely knocked out the pathway of nicotine biosynthesis by knocking out simultaneously 6 genes in the plant. Is something that is very difficult to do with traditional breeding techniques. And in this way, the plants, instead of producing nicotine, they produce another alkaloid that is anatabine, that has added value for other purposes, like anti-inflammatory purpose, and so on. So you can see how you can convert the resulting plants, and these are field trials, do not produce nicotine, and instead they produce anatamine. And we can continue accumulating more knockouts, more mutagenesis, up to create more than 20 genes, and then we have these monster plants. They do not flower in the field, and they accumulate anatamine. They do not flower, and they have higher levels of biomass. And this is a promising strategy in order to have high biomass tobacco bioplants that produce added value compounds. This is one of the lines of research in our lab. But I would like to give you a wider perspective of how these systems are evolving in and the new techniques that have been being incorporated to these new editing techniques or new breeding techniques. We have new what is called base editing. Sorry, this was in Spanish, but well, it's very easy to understand. We can introduce breeding without breaking, and there are two alternatives now. One is this base editing approaches, that instead of producing breaks in the DNA, what we do is to produce nicks and then change bases individually, base by base. This is the equivalent of changing in an instruction, in a genetic instruction, to change some letters, and this produce a more precise change in the DNA. I put here an example of what can happen if you change only 2 letters in the DNA instead of producing a completely nonsense sentence. What you can do is to change slightly the sense of the sentence, and this is what the base editors do. And this can be used in breeding. For instance, here I put a few examples. It's not from our lab, they're from other labs, although we are producing also using base editors nowadays, or nitrogen uptake, dwarfism, and many other precise additions that we can take advantage of in order to improve our crops as well. And then the final, probably the most advanced or the most promising nowadays of these systems is what we call prime editing. What it does, prime editing, is take advantage of CRISPR-Cas nickase. Well, it's a bit complicated, but also another enzyme that is called an RNA polymerase, RNA-dependent RNA polymerase. And what it's able to do, this technique, is to precisely change a few letters in a few words within the genetic instructions, but very precisely, exactly what we want. And in that way, we can change more dramatically, but also more precisely, the genetic instructions, and create more precise or more subtle changes in traits. Like, for instance, I think something that is very interesting, that is generation of new allelic variants, simply changing small sequences in the promoter of certain genes that have large pleiotropic effects or large influence, for instance, in the size of the plants, right? And then quickly, I will finalize talking about cisgenesis and intergenesis, are other new breeding techniques in which we can introduce pieces of DNA from species that can be crossed with our crop species, right? That are what is called, they belong to the breeder's pool. These are also very interesting techniques that are within the basket of new breeding techniques. And this has been for a long time used in order to introduce, for instance, gene resistance from varieties or cultivars that are resistant into some of other crop cultivars that, for instance, in trees, in apple trees, that help in order to obtain resistance and to transfer resistant genes from one species to another, particularly in those cases in which we cannot rely on sexual crossing. Or in our case, also in our lab, what we use is this intergenesis system, in which we combine different parts, different fragments of DNA, all belonging to the same species. In this case, in tomato, we do not take any sequence outside the tomato genome, but we simply shuffling them in a let's say, smart way, and in that way we can get biofortified tomatoes that produce much more antioxidant flavonoids, more than 400 times some of antioxidant flavonoids, and also are rich in branch amino acids that are also interesting for certain conditions in human conditions. And well, that's all what I wanted to tell you, to give you a view of the system, of what is going on. Only later on, if you like, we can discuss a little bit about how is the legislation that is quite difficult quite changing in different parts of the world. And this is my laboratory, and many of the works that I was, the work in our lab I was explaining is made thanks to all these people, in particular in collaboration with Tony Granell. And that's all.
Thank you very much. Thank you very much, Diego. Okay, so the floor now is for Erika.
I completely changed the topic. I'm going to talk about temperate fruits and how can we phenotype the agroclimatic requirements in order to adapt to the challenges of global warming. Oops. First of all, I work at CITA, is in Aragon, here in Zaragoza, and it's a research center that depends on the regional government of Aragon. When I'm talking about temperate fruit trees, I talk about stone fruit as sweet cherry, apricots, and plum, but also as pit fruits as apple and pears. Just sort of reminder, what we see in an orchard are clones, and so just one individual, or sometimes we perhaps see two to guarantee pollination. But and the process to produce a new cultivar in temperate fruit trees is, let's say, quite simple. You select the parents, then crossing, then you put in the field all the offspring, phenotype them with different techniques. You can do a first selection, and finally you select the cultivar that you really want. This that is, well, let's say, quite simple. It can take in temperate fruit trees at least 10 years. And if you wanted to phenotype characters as flowering or fruits, you need to wait the tree till the tree overcome the juvenility. So this is at least 4 years. So we really need in temperate fruit trees advancing the way of phenotyping, early phenotyping in characters that we can identify with well, in genetics, and that we really have the markers to facilitate and so on. So what's happened with temperate fruit trees, how we can adapt a crop that is in the field for years, or at least 15 or 20 years in the field, and to face the challenges that global warming are showing us. One of the main, one of the important effects that is having the rising of temperature is the decrease of chilling accumulation during the winter. It sounds we are like extremely worried about a heat wave in the summer, but the loss of chill in winter is really threatening the viability of these orchards in the Mediterranean area. This is because the temperate fruit trees synchronize the phenology to the seasons. They flower in spring or late winter, and to have this flowering and fruits during the suitable seasons. But if we look but to survive to the let's say the bad conditions during the winter, they enter into a dormant state. During this state, they need to accumulate or to be exposed to low temperatures to be able to bloom with normality, let's say, to have a good bloom and being able to develop fruit in a spring. And one of the main limitation is we don't have tools to phenotype this. This is a character that is genetically regulated and is cultivar specific, but difficult to phenotype. To see how global warming is affecting winter till. This is the evolution of winter till in Zaragoza, this is a cold area. And we are having, and the till portion is the unit that we use to calculate the accumulation of till that is affected for the trees. And we are having significant decrease of winter till. If we pay attention to worldwide predictions, if you see here, just mention that, look at the Mediterranean area, the winter till losses that are predicted are extreme. Here in this work, what we did in collaboration with Moroccan and Tunisian colleague, if you see the loss of till in these countries in the north of Africa, reach 40 till in portion, that is almost all the till that they accumulate during the winter. So this means that these crops are not longer able to be possible to be cultivated there. And they are now facing this problem as well as in Spain and in the south part of Spain. And here in the Ebro Valley, that we are like quite in the north part, we are going to, is predicted to lose by the end of the century between 20 and 30. That this means that all the cultivars that are growing now, they are not going to be viable in the future. So what's the main problem is that the till accumulation, once the buds are able to grow, there are not any change. And we really know little about the physiology of this process to detect when they have accumulated enough till. How do we phenotype this character? We use like quite old techniques like sampling shoots, putting them into a growing chamber and evaluating when the buds are able to respond to this worm treatment. And then we can see that this tree have accumulated enough till. There are other methods like statistical methodologies that we analyze long phenological records, let's say flowering dates of 20 years that we relate with the temperature during the winter. And then we statistically analyze and we establish the period of till and heat accumulation. Once we have established these requirements for flowering, we use different models to quantify the temperatures, the till and the heat. Currently, there are very little information on the agroclimatic requirements of the current cultivars that are cultivated, and my work now is working with my team is on phenotyping the new cultivars and also predicting the adaptability to future conditions in different regions. As we see here, these cultivars that are flat, let's say flat pitches, will behave well in 𝐂𝐥𝐢𝐴 in the future in Zaragoza, but in Badajoz, some of the cultivars will face some problems in the future. And yeah, and now we are trying to offer information about this character in the available cultivars of the different crops. But what happens with when you wanted to develop a breeding programs with this taking into account this character? The method I described, they are not useful. In one hand, the empirical one requires a lot of plant material, a lot of labor, infrastructure. It's not feasible, and the statistical one, let's say we can wait 20 years to get to have a phenological data set. So what's other part, what we are trying to approach is trying to understanding a little bit more about the biology of this process. And we are and also looking for biomarkers in order to see if there are some indicator that could give us a key and to characterize, to phenotype this trait. I work with flower development. It's a very well-known process that's occurring in parallel and closely related with dormancy. So a success story is that the male meiosis in apricot occurred just after till accumulation. So it's a very good biomarker to characterize this trait. And is, well, we have validated with different techniques, and so it's ready to use, and we are applying that in for apricot cultivars. But it's not a universal, and different species have different synchronization of the flower development with dormancy. Other candidate that we are working with is the stats accumulation in flower development. Well, stats accumulation on the placement occurs all along the flower development, and we are trying to relate this with the temperatures that occur during dormancy. This is a work in progress, trying to develop a technique to easily characterize these stats waves in order to find a character related with the chill accumulation. And well, and we are also keeping open, well, we keep our eyes open to see, looking for more processes and that could give us an idea of what's happening during dormancy in temperate fruit trees. And yeah, that's well, and I know this is like quite a descriptive word, but it will be great. There are a lot of information on the, well, there are a lot of works on the genetic regulation of dormancy in temperate fruit trees, but there's not a let's say a ready-to-use tool or a really knowing pattern of what's happening and in terms of the genetic regulation of this trait to really apply genetics tools to know how they are going to behave under different winter conditions. And yeah, I would like just to give some take-home messages, as that the global warming is threatening our survivability in the Mediterranean basin, and one of the solutions to guarantee the temperate fruit production in the Mediterranean, that is they're really important crops because of their high added value, is breeding programs and genetic. But we need to improve the how to phenotype the agroclimatic requirements in order to predict the capacity to adapt to future climatic conditions, and also go deeper in the understanding of the biology of dormancy to easily phenotype this trait. Thanks for your attention.
Thank you very much, Erika. Erika, thank you very much for the presentation. Okay, so we end with the last presentation from José Miguel. Go ahead, José Miguel, please.
I will talk about more than breeding techniques, the most important in breeding that I think is the plant material. No, so I will talk about the genetic resources that for the next generation cultivars. In the 2 years ago, Agata Gadaleta from University of Bari and me were invited to write to edit a special issue in international molecular science about the new breeding technologies in grasses. So we had very few papers, most of them talking about GWAS or genomic selection, but the most important that we could understand from the papers is that the new breeding technique report an economic advantages to obtain a new cultivar, as the time this time is irreduced, and also this technique allows breeders to introduce target traits in efficient and a precise way. Then they can accelerate the selection procedure. So it is the meaning is that they don't have to spend too much money as in the previous years. So to develop a new variety, we need essentially three things. The first one is variability that is related to genetic resources, but the other ones are the capacity of detection of this variability and the capacity to manipulate and develop a new cultivar belong to the new breeding techniques that the other colleagues has explained better than I can do. So here in this graphic, what you can see is that from the wild relatives to the improved cultivars, we improve the agronomic performance, but at the same time, the genetic diversity has been reduced in our cultivar. So this starts at least in cereals from the 60s with the Green Revolution. That at that time the world need to increase the global production because of the hunger that the world suffered at that time. But now we are in another scenario. We are in a climate change scenario, and then we should to come back to wild relatives or land races to recover these lost alleles that we lost during the breeding process in the last 20, 30 years. How can we recover these alleles? No, as you can see in this picture, we have the wild species with high variability. Once when the cultivars were domesticated, they lose variability till the modern or commercial cultivars that have only the most important traits, especially for quality or for grain yield. No? Then one of the means to one of the ways to increase this variability in the cultivated crops is to cross these cultivars with other wild relatives to increase or to have these lost alleles in our cultivars. But why then we have to work with genetic resources? So it's clear because they are source of alleles that are very useful for breeding that has been lost during the process. And also we can use these populations because of their variability or their high variability make us the very good population for genetic studies, no? To find new genes or new alleles from known genes to improve the cultivars. And in the case of land races, they are very well adapted to the regions of origin, no? And then we can try to get this adaptation gene from one part of the world to introgress in a commercial variety in another region. So I will use two examples about this the introgression of genes from wild relatives or from old cultivars, and the first one is the 𝐸𝐸 durum wheat panel of Mediterranean land races. I'm novel in the university. I have only 1 year and 2-3 months here in the University of Lleida. Before I was 9 years working in IRTA, just in the same campus, in the field crops program. Then we worked, or my work was mainly with land race collections in durum wheat and bred wheat. So here this is the IRTA durum panel. So it is about 200 land races. It's a small panel, but what the people in the group, because it was developed before I arrived here, they try to cover all of the climatic variability around the Mediterranean basin. They focus only on the Mediterranean. So, but why were land races abandoned for breeding? As they were grown for several millennia, they evolved and mixed through natural and artificial selection and become a source for the biodiversity for different traits, morphological, phenological, quantitative, and also qualitative traits. And they began to be abandoned from the commercial fields and replaced with the new semi-dwarf cultivars from the Green Revolution that were more productive, but at the same time were genetically uniform. So land races have been restricted to growth in low input and organic farming fields because they have several traits that are not suitable for breeding, as the plant fade, they are usually lateness, and their harvest index is very low. So in the work that we performed here in IRTA, we wanted to determine if there was an effect of the prevalent climate of the zones of origin of the land races in the agronomic performance when they were growth in a Mediterranean environment. In this case, the Ebro Valley in Lleida. No? We took different climatic parameters from long-term climatic data from the countries of origins of our population, and then we divided the analysis into periods, from sowing to anthesis and from anthesis to maturity. As here in the Mediterranean region, the most critical period for cereals or for winter cereals is the grain filling, because it's when we start to have higher temperatures and lower rainfall. No? And this is what indicates here these graphs. No? We can see the different, the minimum, mean, and maximum temperature during the 6 years of the experiment, and the rainfall pattern. As you can see, is very clear, or is a common pattern for the Mediterranean. With we cannot predict exactly the amount of water among years, or even within a year. So here, what we can see here is 2B plots with the climatic variables of the countries of origin of the land races. And then here in this plot, what we can see that the first component, the first components explain about 80% of the total variance. We have a clear differentiation between the positive side with the temperatures and evapotranspiration and solar radiation, in contrast of the negative side that are the vectors for rain and relative humidity. So when we plot the different land races from the different countries, we have here a clear pattern from the east of the Mediterranean to the west and the Balkan areas. No? So then we could establish 4 climatic zones, from zone 1 to zone 4, based on these variables. So this is the map of the land races with the different climatic zones, and we can see the different variables. Temperature is increasing from zone 4 to zone 1, for example. The rain is in the opposite. And here we have our agronomic parameters. These two entities: plant height, number of spikes, number of grains, grain filling rate, and thousand or grain weight. So there are some parameters that increase from the warmer to the colder areas, and other parameters it's in the opposite. They decrease from the colder to the they increase from the colder to the warmer. So as we saw this difference, we concentrate only in two parts of the Mediterranean, the eastern one and the western one. We include here land races from Spain, Italy, France, and Portugal. So this is our we are from Spain, so we are more interested in to breed new cultivars for our region. But as they were more different from the Eastern Mediterranean, so we use only these two regions of the Mediterranean. So here there are some statistical analysis, the ANOVA, that were significant for the year, for each subpopulation, and also for the interaction, the genotype by environment interaction, especially for yield and for number of spikes. So we saw that the means were also different among the both regions, and we use here yield and later number of grains and grain weight. What we can see here is that the land races from the east of the Mediterranean placed on the B plot were with in the direction of the water input from sowing to anthesis, and on the opposite the western ones that fit with the water input of the rainfall from anthesis to maturity. All of these experiments are in rainfield conditions, are not irrigated. But we wanted to see if there were differences in the yield formation strategies between both sides of the Mediterranean. What we can see is the eastern land races. For the number of spikes per unit area, they were on the side of the water input showing anthesis, and the west land races were more use better the water available after anthesis to fill better the grain. Oh no, not to fill better, to develop more grains. In the case of grain weight, it is more clear. We have in the West Mediterranean less number of grains, but they feel better during after anthesis. So they use better this water on the soil after anthesis than the Eastern Mediterranean ones. So the next that we have to do is to do association analysis with this panel. We perform for these 3 years field trials a GWAS, and then what we can found were that we had different markers for both subpopulations that are more specific for one of the subpopulations. Here we are not talking about climatic zones, if not genetic subpopulations. And it is what it is shown here. This is in red are the eastern one, and in blue the western ones. So developing this is the regression equation for yield. What we can see that for the Eastern Mediterranean, most of the yield was related to the increase of the number of grains. But when we saw the Western Mediterranean land races, we saw a more equilibrated balance between number of grains and grain weight. We thought that it is in this case it is more balanced because we use mostly of the zones 2 and 3 instead of the zone 4 that was the most colder and water region. And here this graph it is only to represent that for each one of the traits we have in our population the western allele for the western land races and the eastern allele for the eastern land races. So we can use these alleles to follow a marker-assisted breeding approach for to develop new varieties in the breeding programs with these traits of interest. Another example to integrate genes from wild relatives is the use of near isogenic lines or the introgression libraries. This is an introgression library developed in the University of Bologna by Silvio Salvi, and I was working in this population during 2 years with him in Bologna. And in this case, they introduce different flowering time genes from gaspeflin in B73 cultivar. That is a common commercial cultivar, and the cultivar where the genome was sequenced, no? Or the reference genome. So this is the process of all of the crosses done to develop this library. First, the first cross, and then they start to back cross with the commercial parent. But then, when they have most of the genome introgressed in B73, they start to self these lines to try to get homozygous plants. So this is the final introgression library. Here they are represented the maize chromosomes, and here the introgression lines. And these are two traits that were used for mapping the number of nodes that indicates the height of the plant, and also the days till anthesis. So in this case, it is very fast to identify one QTL and then to identify just the line that has this QTL and the small region in the genome that when we can have these genes, or even to develop or new breeding lines for a particular line. So how can we integrate these genetic resources with new breeding techniques? So as I thought that someone will talk about speed breeding here, but at the end nobody did. But we can consider speed breeding as a new breeding technique. We can develop different we can have different several cycles of progenies in a year. In wheat, they arrive till 5-6 generations in 1 year using speed breeding. Also, what two of the colleagues talk, we can modify quickly candidate genes using the gene editing or base editing, as Diego told us, and the use of genomic selection. That is a new breeding technique from the last 10 years that allow us to identify without phenotyping the better breeding lines for quantitative traits, as mainly all of these studies are performed for grain yield. And now we are introducing also the accurate phenotyping using a remote sensing platform that for quantitative traits will be very useful to not to spend too many time in the fields, and also to analyze in one picture or in a small period of time different traits that now we can use 2 or 3 weeks to analyze a large population. So then in that case we will have a picture for a single moment for all of the population, and it can help to homogenize the results for the phenotyping. So now the most important is, as this triangle is shown, is to integrate the three vertices: the genomics, environmics, and phenomics, no? And to to reduce field testing cost, the computational demands, and also to improve the genotype by environment characterization. So thank you, and I hope to see all of you in 𝐸AMP. 𝐸AMP is this institute in the Aula Dei campus. This is the CSIC, Aula Dei CSIC, and this is the CITA. But 𝐸AMP is this part. So thank you very much.
Thanks very much, José Miguel, for your presentation. So I think we have time for some questions. There is in the chat. There is no for the moment no question, but I can launch one of the questions, the most easier one and most complicated one. So, what are the main technical challenges or limitation of new breeding techniques? So one of you can answer, whatever, Diego, Elena, or Erika, whatever. So what are the challenges or limitation of new breeding techniques?
Well, I think the one I can agree that regulation is probably now, even we can do more things technically than can get approved and be actually used in the field. I don't know. I feel this is the bottleneck right now.
Yeah, José Miguel was very right pointing out that when we talk about, we are a little bit perverted to think about new breeding techniques, only the new genetic techniques that we were focused on. There's some other new breeding techniques, and we are very molecular, and we focus in the new legislation and new regulation about new genetic techniques, and particularly for those breeding techniques that relate with new genetic or genomic approaches, I think regulation is the main problem. There are certain regulations and also to a certain extent, I mean, when it comes to crops and to applying, and again, I'm focusing in these genomic techniques, the NGTs, also the ability to transfer DNA or transfer new genetic information and to fight against recalcitrant species in crop species for regeneration is also a technical challenge nowadays.
Erika, something to add or not?
Yes, I was thinking on improving. Well, the first thing was regulation, but I was also thinking that phenotyping needs to be improved and make it easier. So phenotyping, we need to, let's say that it's easy to breed when we have like well characterized the traits and we know what's behind, but in some characters as adaptation and other quantitative traits, it is needed to improve the phenotyping, the efficiency of phenotyping, because otherwise it becomes really complex to so it's easier to have advance in some traits, but in others it becomes extremely difficult. So
I agree with Erika, especially for gene isolation. We have large populations, so it is easy to have now thousands of genetic markers. But if we don't have good phenotyping, we will not look for in the right region. So probably it's more important the phenotyping than not probably it's more important than to have an accurate phenotyping than a genotyping.
Yeah.
Yeah.
Okay. Yes, go ahead, please.
No, I was saying that this new platform for weeds that are able to, with new cameras and that they completely control the environmental conditions in the greenhouses, and they are like recording images and taking like live measures, are like improving a lot the phenotyping in weeds. In weeds, or in extensive crop, I mean like wheat or well, cereals, and I think that this is a big advance, but yeah, for other things, we need also to keep on combining like basic science with more advanced approaches.
Okay. So what I think, what the people, when we talk about new breeding techniques, the people that think that you are talking about all the time about GMOs, And this is not a reality, but the people that think what new breeding techniques, GMOs, whatever. So my question is, Lancet, for you is what lesson can be learned from the regulation of GMOs that could apply to new breeding techniques?
Yeah, if I might start.
Yes.
Again, focusing in new breeding techniques that relate with genetic modifications. Well, there are a lot of lessons to learn. I mean, GMOs were regulated in the EU, in other places very strictly, in the 80s, in the 90s. And this came with a, from my point of view, with a delay in the possibilities or exploiting the possibilities of doing this kind of biotechnology for the good of humanity. And I think we are learning from those mistakes, and now that we have a new opportunity to introduce new modifications in the genome, because at the end what we do always is to introduce changes in the genome in order to do breeding, and this is unavoidable. So I think we have and that's what authorities are probably doing, is to ensure that all the safety requirements are met, but at the same time balance also this with the possibility of technical advancements, or incorporating technical advancements in agriculture. So I think the right way is to have the balance between the two points.
Thanks, Diego. Yeah, Raúl, go ahead.
Would you give me the floor?
Indeed,
Can I ask
yeah.
to our speakers, to our
please.
experts, Diego, I don't know who, feel free to answer to me. My question is, do you see a change in the priorities, I mean, in financial resources, in budget for research in this field? Do you see a change in the institutional, in Europe mainly, regulations due to the crisis? I mean, do you have, all of you presented the challenges? All the challenges are connected more or less with the new conditions, bad conditions, climate change, we can discuss or not, but water stress, mainly if now it's raining a lot of in March in Madrid mainly, and so on. But looking ahead, do you see more conscious in the political and institutions concerning with these topics in order to give more resources to research, or to open the field for new techniques in order to respond to these challenges, or not? Or do you see the same mentality, the same resources? Because my position, as I am not a specialist as you, is that at least in the visits that I do in several countries, I am appreciating a new sensibility to watch vegetal and animal breeding too, in order to respond to the new challenges, even in terms of food security. Even if we don't think in terms of climate change challenges, I see a more disposition to open to give resources. What is your point of view?
Regarding resources and Europe, I think yes, it became a focus clearly in the last programs of Horizon and all that stuff, the fight against climate change. And I think in terms of Europe also, like we were talking before about regulation, it's clear that they've also learned the lessons from what happened with GMOs, and it's been hard, and there's been a lot of talk, and probably the agreement they've reached is not great, but at least most countries in the EU have agreed on a less complex and less problematic way of getting edited crops into the market. So I think, yes, the frame of mind in general is more like understanding that this is something that we need to do in order to face the future, and in terms of, yes, food security and whatever, survival even. So I wish it was even stronger, and more resources were allocated here and there, and more permissivity to products that are generated through NGTs that could be generated by crossing in maybe 1 century, and we can now do it faster so this would be easier to get into the market for the citizens of Europe to enjoy. But at least it's something. No? So I think the trend is positive. I don't know about the other.
What do you think, Diego?
So more field, more floor for this kind of research and studies and training, isn't?
May I add something?
Yes, go ahead. There you is.
Yeah.
Yeah, I fully agree with Elena that we have seen this interest that is probably driven by the fact by the need of the realization by authorities and from many stakeholders that genetic breeding is probably one of the most valuable answers or responses to the current challenges, and then we have to open the floor to the best technologies we have, and this is something a realization that this is has been during the last years, and I really will hope that it will continue being like this. To first to a relaxation in the requirements of in the regulatory, and also we see in the European programs and other programs to more and more interest in funding this kind of research, and also very importantly also from the private sector, because they see that for the first time that this kind of techniques can also be applied and have a horizon of commercialization. Having said that, I'm a little bit afraid in the last 6 months or even less that the focus is changing towards other priorities in Europe and in the rest of the world, and I'm a little bit afraid in the recent days that defense and self-sustainment and so on will be diverting some of the efforts that we are seeing now in breeding or in biotechnology, and it will move to other fields of research that are more related with war than with food. But anyway, let's hope this is not the case.
Thank you. Sorry, Rancy, there is a question for Erika. Has one in the chat for Dr. Erika.
Yeah.
A question. Have you read?
Yes. Yeah, in the question and answer panel, Hasuna, I don't know if I pronounced
Hasuna,
well.
yeah.
Hasuna.
Is regarding deficiency use.
Well, the histological approaches were very useful in the case of the stat to identify the different dynamics and to see what's happening in various small tissues, but it's not feasible for really a fast evaluation of the chilling satisfaction or fulfillment. So we are developing another techniques to measure this in a faster way in order to do it more efficient. I don't know if I.
Okay, I have another question, and I have to end because of our time. There is another question about how can we utilize artificial intelligence tools to enhance new breeding techniques. So, do you have any idea about that?
There are certainly, if I might start, there are certainly many examples in which we can do this. I can give you just a simple one that comes out of my mind at this moment is that for instance, nowadays there is a big controversy and difficulty with the CRISPR-Cas patents and licensing and so on, and now so who owns the patents and all the CRISPR-Cas tools? And now suddenly it appears that we can start to think about designing de novo new CRISPR-Cas enzymes that have the same activity and that are not related whatsoever with the original ones. And all this ability that probably you heard about AlphaFold and all these tools that we can start designing, and there are some examples now of de novo design of enzymes that open the amazing capabilities to create new tools in the more molecular side. So probably there will be many others, but this is just a simple example.
So while we are at the end of our webinar, thank you very much. So as we wrap up today webinar, let's take a moment to recap. The main insight shared by our panelists is we have explored the transformative potential of new breeding techniques in agriculture, highlighting how these innovative methods can significantly enhance crop productivity, resilience, and sustainability. So before we are leaving, I would like to express our deepest gratitude to our distinguished panelists for their insightful contribution, and to all of you for your active participation and through full questions throughout the session. Your engagement truly made the webinar a dynamic and valuable experience. So thank you once again to joining us today. We hope we found this session enriching and insightful. Stay connected and continue exploring the exciting world of new breeding technology. So thank you to everyone, and hope to see you next time, while here in Zaragoza or whatever in this life. Okay.